Hi everyone, we're really glad to be joined today by Julie from the University of Birmingham, who's going to share her research with us. So Julie, thank you so much for joining us. Can you tell us a little bit about your research?
I'm interested in looking at clotting under infectious conditions that could be in sepsis as bacterial infections or in viral infections like COVID-19 patients, and how this clotting is occurring in different organs, and looking at different ways to stop this clotting that is different from the clotting that happens after injury. What we really focus on in my group is to understand how clotting happens mainly in non-classical thrombotic conditions like infections, like hemolytic diseases, and how these mechanisms are different from classical injury that can induce clotting. So we focus on bacterial infection, and more recently we try to understand how the clotting happens also with COVID-19 patients. And a big focus, which is also my BHF Intermediate Fellowship, is how hemolytic diseases and the crosstalk between red cells and platelets alter the interaction of platelets with immune cells and the endothelium, mediating this inflammatory environment and a thrombotic environment under these conditions.
So that sounds really interesting, Julie. So we know that thrombosis is a problem for people that have COVID-19. So how do you see the potential impact of the research that you're doing now to help us better understand COVID-19 and associated problems?
The one thing that we realize now with COVID-19 is not a very simple clotting issue. So it's a complex disease where there is multiple factors that are leading to thrombosis. It's not, as we know, patients do not have a major platelet activation issue, and this is possibly secondary to something else happening, and which possibly we think is inflammation. And this COVID-19 is one of the examples where inflammation is driving clotting, rather than a vascular injury driving clotting. It is very complex, and as complex as we know, the treatment will be complex and it will be multifactorial. And also it will depend on the stage of the disease. At the early days, we'll have a hyperinflammatory response that will require a treatment, whereas later, where the immune system is driving possibly the clotting, will require another treatment. So what we can say now, that this is very complex. The research and the basic research is really important to understand what is really happening, to try to identify a treatment that works at each stage of the disease, which one could be beneficial at some stage but become detrimental at another stage, and possibly find a biomarker related to which drug could be used at this stage and could be beneficial. And I don't really think that one drug would be a yes or no treatment for COVID.
So you also mentioned that you're interested in looking at clotting and thrombosis in hemolytic diseases, and I wondered if you wanted to just expand on that a little bit more, because I think that's probably a very understudied area, particularly by the platelet field. So I think you know that's your niche there.
So the role, the clotting under hemolytic disease is the basis of my BHF Intermediate Fellowships. And when we talk about hemolysis, it could be just genetic disorders like sickle cell disease or other genetic disorders, but it also could be in infections like hemolytic uremic syndrome, sepsis. All these diseases and infections really induce red cell lysis, or by mechanical destructions, or also by muscle injury releasing the myoglobin and heme from the muscle. So there's different contexts where this kind of hemolysis and myolysis can happen. And then what we know that is associated with thrombosis, what we don't understand why these hemolytic diseases and trauma and then muscle injury can lead to this thrombosis. So my aim is to understand what's the role of platelets, a platelet activation that we know is occurring in hemolytic diseases that could be congenital or in infections, and how can we prevent it? And then looking at specifically the interaction, interaction of platelets with the innate immune cells that seems to be regulating vascular integrity and thrombosis, and regulating endothelial function. So it's really a crosstalk, looking at the crosstalk between the endothelium, platelets, and the immune cells in driving this inflammatory and thrombotic environment in hemolytic diseases.
So it sounds like you're approaching kind of the thrombosis from a slightly different angle to other members of the Platelet Society, perhaps. Just wondered if maybe there are any techniques that you use that you think maybe other society members should be perhaps using a bit more, or
so as looking at infections and hemolytic diseases, the in vivo experimental models are the basis of our work. So we do very little in vitro work, and then the in vitro work is mostly to dissect the mechanism that we see in vivo. One of the looking at the most techniques that we use is very complex flow cytometry, because we're looking at different populations, and then this is basically intravital microscopy, which is very important, because when we look at the cellular level or at very quick mechanism, this is the basis of what we really do. Mostly flow cytometry, intravital microscopy, and a lot of tissue collection, sectioning, and staining when we look at thrombosis or inflammation. A bit of really everything, lots of cytokine measurements by Luminex or by ELISAs. We do a little bit less than what the Platelet Society would do, like aggregations, and because we look at in vivo mostly mechanisms. So all the in vitro assays that we do are mostly on to dissect specifically a mechanism rather than doing a full studies in vitro. And so I would say intravital and flow cytometry would be the techniques that we use the most. And I do think when we look at acute inflammation or acute thrombosis and looking at interplay between different cells and the endothelium, we do realize that both the intravital and then the complex, like multiple colors flow cytometry, gave us a lot of information that we would have missed if we were looking at much smaller scales of flow cytometry, for example.
I think that's a really important point as well, isn't it? Because I guess by collecting the tissues as well, or imaging it in real time, you actually kind of get to zoom out a little bit, don't you? Kind of see, okay, so we have thrombosis, but also what's the effect on the surrounding tissues? And I think we tend to think of platelets in isolation when really they're in a very complex environment. So as we know, research is funded by all sorts of charities and councils in the UK and around the world. We're just wondering how the work that you're currently working on is funded.
I have different sources of funding currently, and it's mostly through PhD studentships. So it's part funded by Compare, which is the microscopy center between University of Birmingham and Nottingham. Wellcome Trust funded a PhD student, and I do have small grants from the BHF to support this work, and also from internal funds from the University of Birmingham. And also I recently got a BHF Intermediate Fellowship that will support further the next few years of research.
So congratulations on the BHF Fellowship. That's amazing. We were hoping that you would be willing to maybe share how you found approaching writing the fellowship, putting the fellowship together, and if you've got any tips for members of the society that are considering or planning to write their own fellowships.
The one thing that I would like to say, that this is not the first fellowship that I write. So it's not like the one go fellowship we write, we get it funded with no revision. So it was revised. It wasn't funded from the first round. So I would say don't get disappointed if you submit a fellowship and it gets rejected and then you work more on it. And sometimes, so I did put before another fellowship on a different project that wasn't get funded. And I think sometimes we just realize this is not gonna work. And sometimes we just have to accept that we have to change sometimes ideas or look at it from a different angle. The one thing that I learned that the grants have to be novel, ambitious, but realistic, because I think possibly the first one wasn't very realistic. I think it was, I think these are 5 years fellowships where, you know, you have 1 or 2 persons maximum working on it. And sometimes we write it as if there's 5 people who's going to work on it for like the next 5 years. Because I think we put lots of energy and we think that's going to work. It's very nice, but I think it's just not realistic. And I think that's a mistake that I've done first, to try to have it over ambitious to show that it's very different. I've learned with my second attempt for the intermediate fellowship with the BHF, because I've looked at something that it was novel for me. There was lack of information in the literature. And it's to understand that there's the project, the person, and the place. The 3 of them have to come together. If there's an ambitious project but not in the right place, and there's no support and structure to support it, it will fail, just not because the person couldn't deliver, just because there's no environment to support it. And I think what they say, the 3 Ps are true: the person, the place, and the project. So I think find the right place, find the right support with the right project, and if you're the right person, I think that has much more chance than if one of them is missing. I think it would be more difficult. Another suggestion is send it to friends who you think are really friends, who can read it and give you advice on it. Sometimes I do say, kill my grant, and they do kill it, and you come crying after, like, is it that bad? It's not that bad, but ask them to just be as hard as they can on the grant. So then if you can address all these comments, you know, when it goes after for reviewers, you've already addressed a lot of the concern that anyone could have. And I do think it's very important to send it for people to read and see their comments, because sometimes we're on our bubble and we think that's it, it's perfect. And sometimes someone comes from outside and pop this bubble, and I'm like, oh, I didn't see that. So it is very important to have also some support, and then collaborators, friends, find the right collaborators and the right friends who can help you in this. We can't do it on our own. We just need some support around us to make it happen.
So we were wondering if you know, what's your journey? How have you got to where you are now? Would you mind sharing that with us?
Yeah, sure. So my journey is not like as straightforward as as many people maybe. So I have a master and a PhD in thrombosis and hemostasis, and then I wasn't sure always that academia is what I wanted to do. So after my PhD, I worked for 1 year as clinical research associate at the hospital in Paris, where it was mostly for a bit of clinical trials, and then understanding in the clinical side of the research that we do. Done this for 1 year, which I realized that that's really cool, but maybe not what I wanted to do. So I went back to do a postdoc the same lab where I've done my PhD, just because I wanted to, it was developing a new experimental model, and then I've never done this kind of work before, and it was a great opportunity. So I've done 2 years, and again, after these 2 years, I wasn't sure that academia was what I wanted to do. So I went for 1 year where I've done more teaching at the Faculty of Pharmacy, and I had a maternity leave in between. So thinking of what I wanted to do, because the one thing I wanted a job that I enjoy. It wasn't about only getting a job. It was a job that I knew I'm going to stay for the next 30 years doing it, and I wanted to make sure that this is what I wanted to. And after that, I realized what I enjoyed the most was really research and working in academia. And then I've decided, well, now I'm going to work toward getting a job in academia. So then the one thing that was I had to do is to learn something new. And the one option that I wanted to do is to go to immunology and really understand mostly the innate immune system and inflammation, because there was this crosstalk really coming from the coagulation side and the endothelium and inflammation. So I did 3 years postdoc in Paris at the Centre d'Recherche et Cordelier in immunotherapy unit, working on innate and adaptive immune responses to clotting proteins and how the immune system interacts with the clotting protein. And this is where I realized I think this is what I would like to do. I would like to work in the crosstalk fields, looking at how the inflammation and platelets and the clotting system work together in complex diseases, because we mostly study it in separate angles, looking at the clotting alone or looking at inflammation alone, but we rarely think that these coexist in the same organ in the same place and they influence each other. And this is where I moved to Birmingham 6 years ago now. So I moved to a senior postdoc with Steve Watson, and I had the chance to develop my own project with Steve, while looking at the inflammatory functions of platelets, looking at my background with inflammation and my interest in this crosstalk, which it was a very nice opportunity working in platelet group and coming from inflammation background, where I could just bring my expertise and develop new things. Last year I got a lectureship at the University of Birmingham, and to also understand this crosstalk between inflammation and thrombosis, mostly in infection and hemolytic diseases. And then I recently got the Intermediate Fellowship. So this is my journey that wasn't very straightforward, but I think I took my time to decide that this is what I wanted to do for the next 30 years. And I think I'm very pleased that at least I'm sure that this is what I enjoyed the most.
Thanks for joining us today, Julie. I've really enjoyed chatting with you.
Well, thank you for inviting me. It was my pleasure to chat with you and then to discuss all these points with the Platelet Society.
Join us next time for another installment of Clot Chat.